Yeah, thank you for inviting me. This is my first time to BOSC and to this conference, and it was kind of, I guess, reassuring for me because I feel like I finally made it in the world of bioinformatics to be invited here. So as Peter mentioned, I'm a biologist, and I'm going to talk to you about biology this morning. And I do not come from this world, so I don't know if I have answers for some of the things I'm going to discuss, but I just want to give my perspective coming from the world of biology and talking a bit about what it's like to go from something that is not computational at all to something that is almost purely computational. And I picked a talk title that's a little bit provocative. I don't know if scary is the right word for how I perceive bioinformatics. So I've been playing around with other ideas, ways to capture this. So bioinformatics for biologists can be scary, it can be daunting, it can be nebulous, it's definitely intimidating. I wonder how we can get around this, how we can make it easier for biologists to work with bioinformaticians and computer scientists and help them analyze their data and get to grips with some of the tools that they have to use. And so I want to start this talk with a number, and this number is 2010. And I haven't ever admitted this in public before, but this is the year in which I saw the command line for the first time. That wasn't that long ago. That was only 5 years ago. And I've gone from never seeing command line before to basically using it every day, and going from being in the lab every day to being at a computer every day. And really what I want to get across to you in this talk, and I guess the point of me coming here is really to say that I am your end user. So I'm going to try and give my perspective as, how I perceive bioinformatics, how I've gotten into this world, and some of the challenges which I continue to face on a daily basis, and also what other people who have never also seen the command line before, what they face when they start to delve into this world for the first time. All right. So to give a little bit of background about my research, I work on marine nematodes. So I did my PhD at the Natural History Museum in London. And I spent my undergraduate career, my PhD career, sitting under the microscope and looking at things which look like this. So my research focuses on microbial eukaryotes with a focus on metazoa because of my background in marine sediments. And so my background is nematode taxonomy. These two things are nematodes, if you don't know that. But really, when we're using environmental sequencing approaches, we're sort of expanding out from our taxonomic silo, because sequencing really allows us to look at everything. And so when I go to the beach and I collect marine sediments, I am looking at not just nematodes, which is where my taxonomic expertise is, but all of these microbial eukaryotes, phyla, which are sort of minor phyla, things like tardigrades and priapulids and kinnorynchs. And these things are everywhere. So anytime you go to the beach or go to a field and pick up a hand of soil or mud, there's going to be thousands of these microbial eukaryotes that are writhing around. You can't see it, but they're there, and they're everywhere on Earth. They're very diverse. They're very abundant. Nematodes can be in the millions per square meter if you're going out onto like a mud flat in an estuary. And they make it down to the deep sea, which is where my PhD work was focused. So they really are this incredible system to work in, because there's these little indicator organisms that are pretty much present everywhere. And yet we know nothing about them. They're not represented in databases. And there's some really cool questions to ask about the evolution of these taxa, how they disperse, how they are affected by environmental change, climate change, oil spills, things like that. And so I'm really interested in really focusing on neglected taxa in an understudied habitat, because no one cares about them. So there's my research niche. And to give you an idea of what we tend to do with these organisms, I mean, it's typical environmental sequencing, so metabarcoding, metagenomics. As a biologist, nowadays we're going out, we're collecting samples, we're bringing everything back to the lab, typically doing an environmental DNA extraction. So not looking at individual organisms anymore, but just taking out all the DNA, all the genomes that are present there. Going on to amplify a barcoding gene, typically 18S ribosomal RNA, but you could do metagenomics if you wanted to get a different perspective. And then going on to sequence and, get these biological patterns out of your data set. So looking at taxa across geography and putting that into a phylogenetic framework and getting this nice pretty picture which you can put in your publication, of course. And really that's not so easy, especially for biologists when I'm still struggling with getting to grips on the computational tools. There's this bottleneck from generating your sequencing data to actually going and getting that picture. So this is really where I want to focus my research, and I do tend to focus my research because we have to make it easier for people with the biological knowledge to get their data and then to get that image that goes in their publication and help them use their expertise to really understand what they're looking at with this deep sequencing. So to give an example of that, I'm not going to talk too much about biology. I mean, I could give you a talk on nematode taxonomy for an hour, but that's not really the topic of this conference. So how do we use this? One of the best examples, I think, from my work is looking at the impacts of the Deepwater Horizon oil spill. And this is an actual image of a beach. It was terrible going and sampling on these white sand beaches. This is actually a beach after the oil spill. And if I didn't tell you that, you probably wouldn't know that. They did a really good cleanup job. I mean, they had crews out in the Gulf of Mexico with this massive equipment. They were basically power washing the sand, and they had mechanical cleaners. So the cleanup effort for the beaches was pretty severe, and it was pretty extensive. And if you went to this beach and you hadn't known about the oil spill in September of 2010, which is when this photo was taken, you would have never thought that there was anything wrong with this beach. There had been any sort of environmental impact because the cleanup was that good. And that's where really the power of environmental sequencing was demonstrated, because when we actually sequenced this beach, we found that there was a massive community change from pre-spill to post-spill in terms of the microbial eukaryotes. So the black bars, black means bad. So this is fungi which tend to take over the beaches, what we found anyway in this study. We had samples from the exact same sites before and after the oil spill. And environmental sequencing really let us look at that to a degree that wasn't really going to be possible unless we went in and we used genomics. Okay. So, what am I good at as a biologist? I'm good at a lot of things, and I wanted to take you through some of those things which I have been trained in during my undergraduate and PhD career. So I'm good at collecting samples. So I have had extensive experience going out to beaches, which may or may not be tropical, as well as going out on boats, which is this a picture of. And I think from the biological perspective, which is not really reflected in the bioinformatics world so much, it's really hard to do biology. I'm also a deep sea biologist, and if you had to pick the hardest environment to work in, that's pretty much the deep sea, because you have to pay for a ship to go out, you have to collect samples, deal with weather, and it's just extremely difficult to get samples in general. So a lot of deep sea biology, marine biology, actually goes into collecting your samples, planning out your sampling, and processing those samples, which is a very labor-intensive process, and that's where a lot of our thought goes. So this is just a picture of how we actually go and collect samples. For in the deep sea, we'll go on a boat and we'll send down this giant piece of equipment to collect sediments from 2,000, 3,000, 4,000 meters below the surface of the ocean. So that's hard. I mean, there's a lot of wire. It takes a long time. Typically, you're staying up overnight. Everyone else is at the bar. You're in a 4-degree cold room, not having beer, and wearing lots of warm clothes to get your samples, because those are really precious and you don't want to screw it up. So I'm good at collecting samples as a biologist. As a taxonomist, I also know my worms. So I've spent way too much time sitting under the microscope, working with other taxonomists, learning how to differentiate my worms. So I can tell a nematode from a polychaete. I can tell when something's not a worm. And I know, generally what all these little minor phyla look like when I'm investigating my samples. And then I also know how to draw worms. So I'm really good at looking at these pretty, you could argue, boring, featureless images under the microscope, and using the microscope properly, going through the focal planes to actually understand the structures that I'm seeing, and to interpret those structures, draw them, and then link that with a taxonomic ID, which then tells me something about ecology. So those are the things which I did during my PhD, and none of that really prepared me to use the command line. So I work with a lot of taxonomists and people that are really interested in sequencing, because everyone's heard about that 454 sequencing, and they want to do genomic work, because that's kind of what you need nowadays to be competitive in biology and to write grant applications that are forward-thinking and are going to get you funding. But biologists that have interesting study systems don't necessarily have the computational skills or the understanding of what is needed to really use these type of data and to process them and analyze them in the way that they would do from a taxonomic perspective. So you sort of have similar goals, but you're going about it in completely different ways if you're doing taxonomy versus if you're doing bioinformatics. And one of the things that we struggle with as biologists is really getting to grips with how fast the field moves. So taxonomy has not changed in 200 years, and it's pretty easy to keep up with taxonomy, actually. The difficulty from that perspective is really just learning, training yourself to know what you're looking at under the microscope. So that's more of a personal development thing and not the field is changing around you type of thing. But with sequencing, bioinformatics, everything moves really fast. A lot of what I spend my time on these days is really knowing how I can keep up with the studies that are published. And working on eukaryotic taxa, we're also trying to keep up with the microbial people, so bacteria, archaea, the groups which have really been using molecular data since the 1970s. I still have collaborators that don't even think of doing DNA sequencing or using any sort of genomic data in their day-to-day work. So there's that aspect as well. But to keep up with what's happening, what's the state of the art in the field, we're having to design sampling programs where we're collecting a lot more, so hundreds or thousands, driven by projects like the Earth Microbiome Project, Terra Oceans, when they're really pushing the field forward, but they're making it really hard for the biologist in the corner to kind of keep up and design something. There's also more emphasis on solid sampling design, so making sure we're collecting robust environmental metadata. So that means working with other people who maybe aren't taxonomists, so working with stable isotope people, biogeochemists, other types of metadata that's going to help us understand our target organism better. Also using multiple complementary omics approaches. So doing ribosomal RNA surveys is not going to get you a Nature paper anymore. You got to do genomics and metatranscriptomics and metagenomics, and each of these omics approaches has its own lab protocol. It has its own bioinformatics tools. So you have to do a lot of learning on the fly all the time to understand how these different omics approaches are happening nowadays. And of course, what's kind of worrying, makes you constantly paranoid, is all this bias and contamination and technical error, which is kind of coming out of studies over the last year. And it requires a lot more effort to make sure that you're not introducing this into your workflow, because you want to do biology and you don't want some contaminant to skew your conclusions. So there's less of that concern when you're looking at things physically under the microscope, but when you don't see your organisms and you just sequence everything and run it through a pipeline, you really don't know what's happening to your data as a biologist. So for field work, so from the biology perspective, we're trying to do the best that we can with making sure that our protocols are going to help us do the best bioinformatics possible. So this is even before we get to processing our data. This is what we have to keep up with in terms of physically handling our samples. And so I was trying to figure out how to frame this talk and how to talk about why bioinformatics is scary. And this Donald Rumsfeld quote kept sticking in my head, because I think it sums up my experiences and the experiences of other people that I've helped train in bioinformatics. So as a biologist, when I'm trying to do bioinformatics, there are known knowns. So I know there's things that I know. There's also known unknowns. So there are some things I know I don't know. And there's also unknown unknowns, which are the most scary, and those are the things that I don't know, I don't know. So going through these known knowns. I know what I can do as a biologist transitioning to computational biology. I know I can write a Perl Python shell script. I know I can sign up for training courses. I know how to Google error messages, which is the secret of bioinformatics, I've discovered. And I also know who to ask if I get stuck. So I know I can do these things. I'm pretty confident with my skill set. I know I don't know everything, but if I give it enough time and I Google enough things, then I'm confident that I can figure things out, usually. But there's also known unknowns. One of the things that constantly strikes me is how much there is out there in the computational world that I have no idea what it is. And so you don't want to give off that impression. You want to try to stay cool and try to talk to people. But occasionally they'll throw out words like Hadoop and Drupal and Ruby on Rails. And as you're talking to people, you're sort of thinking, is this an app? Is this a programming language? Should I learn this? Everyone has their opinions. So some people are very adamant about learning certain things. How have I not heard about this before? You feel like such an idiot. And also, God, just another tool. When am I going to have time to read up on this? So there's all these things which I should look into, but I just haven't had time because I'm already trying to keep up on the developments on the biology side of things. And so that's where my expertise is. That's where really my interests lie. I'm a marine biologist, so I tend to read the marine biology papers, and I don't necessarily have time to sit down and understand what is the best way forward in terms of some of these new tools that come into play. All right, and then there are the unknown unknowns. So these are the things we don't know that we don't know. And coming from a biology background, I think this is, this is kind of scary when I have this revelation, this eureka moment that, God, I really should know something. But I had no idea. No one ever told me because I never did a computer science degree. These are things like core computer science concepts. So no one's ever sat down with me and taken apart a computer and shown me how a computer works. Again, lots of Googling, lots of conversations in the bar usually. And I'm sure there's other core computer science concepts related to software that I don't know about, but I should probably know about in hindsight as well. One of the things practically which comes into play for biologists that I train is this understanding of parallel threads versus distributed computing. So how your software runs on a system, whether or not that's shared memory or that's something like MPI. That's important to know, especially when you're trying to work with your local cluster or your, if you have a supercomputer on site, when you're talking to those people, you need to understand how your software is working in order to get it installed properly and make sure that you're making use of the available resources and you're maximizing that use. And then, something which has come into play recently in the QIIME pipeline, which is something I use a lot, is this transition between files. So QIIME recently switched from text-based to binary formatted files, which you don't know unless you look at the file. And surprisingly, biologists don't tend to look at files. They get outputs, and then they try to use that output in another tool, and then all of a sudden it doesn't work. And it doesn't always get across that, even though they're calling it the same file, there's actually been an update in the version of the software, and they've completely changed what the format of that file is. And that's something really people don't tell you. Also scary is this disillusionment that you kind of face as a biologist doing computational work, in that you kind of become this interdisciplinary entity, and after a while you're not really sure what to call yourself anymore. And you could argue this isn't important, it doesn't matter what you do, but it is, because when I'm writing a grant application, I want to know, am I a marine biologist? Am I an environmental microbiologist? Am I a computational biologist? Or am I a molecular ecologist? To a biologist reading a grant application, all of those things mean slightly different things. And it's important to sell yourself. It's important to put on the hat, which is going to make you most attractive for funding or for jobs. And I've had this experience when I'm applying for things, in that if I don't sell myself the right way, it's perceived that I don't have the right skills. If I call myself a marine biologist, then I really should have called myself a computational biologist. That's an issue. But if you don't call yourself anything, you get the you get a response of, well, what do you actually do? Because no one's quite sure. You do software development, but then you also study nematodes, and then I've also done work in the built environment and with bacteria. So, I like doing all those things, and I think that I have this core idea of what I do. But biology and departments and people in science can be very traditional and still not have adapted to this changing landscape of people becoming more interdisciplinary. All right, so from here, biologists who need bioinformatics, I think there's two ways which they can proceed. They can hack through things themselves, or they can use a prepackaged pipeline or pay a core facility to do these analyses. And I think, there's no one solution. I think that either of these solutions it can be appropriate depending on what you want to do with your data and where you want your research program to go. And each of these solutions also has upsides and downsides. And in thinking of a metaphor for how I was going to convey this, I wanted to use an example from my favorite video game and probably my first exposure to computers, and that's the Oregon Trail. So the options for biologists using bioinformatics, you have, I think it's very similar to fording a river in the Oregon Trail. I don't know if anyone's played this game. I know this is an American obsession. But in this game, you had to forge rivers, and you got to make choices. So you could try and forge the river yourself. You could attempt to caulk the wagon and float it across the river. And this is what I would say is similar to a biologist trying to hack things together themselves. You see the river and you're like, oh, I can do it. It's great. I'll just, I'll pick option number 2 and I'll float it across myself. And everything's going great. And then all of a sudden things fall apart. So your script doesn't work. You erase your data, or something happens and you lose things. It's not good. So, you walk around in a heavy fog, you're really upset, you lose a lot of time, and then eventually you have exhaustion. You're just so tired of trying to hack through these things yourselves that option 2, paying someone to do it, actually starts to look really good. So option 2 is to use a pipeline. There's a lot of core facilities that offer options where you can just give them your data and they will hand you back output files that are, just graphs and things that you don't have to do anything to. You can just stare at them, nice visual images. And this is the same thing as taking a ferry across a river. You pay someone, you wait, and you know it's going to work. So you just get on the ferry in the Oregon Trail, go across the river, and everything's a success. You don't have to stress, you're not tired, you just have your outputs ready. And then occasionally, I think this is the perhaps not so common, but the great thing is if you have a Shoshone guide that offers to sit down with your data and explain things, and then you can have someone with knowledge, but you can also get the information, which is not common. I mean, that sort of happens in training courses, but apart from that, a lot of times as a biologist, you're on your own processing your data and trying to figure out what happens. So I'm not saying that I have solutions to these kind of problems, but it's something to think about. How do we get biologists to feel less intimidated by hacking through their data? But how do we also avoid the black box mentality that comes with just handing off your data to a pipeline or a core facility? And personally, I really worry about core facilities and things like Galaxy installations, because it doesn't really teach biologists to understand what's happening with their data, and that's extremely important, particularly for things which don't have very good database representations like microbial eukaryotes. I think it's extremely important to understand how the software is working and how changing parameters can actually affect your data. And really, the only way you would get that experience is by hacking through the data yourself, which is the more painful option, which is not the easy way out. And it's been my experience also that bioinformatics is great if you're an absolute beginner, because there's a lot of training courses that you can sign up for. So there's software carpentry, there's different languages, different programs, and you can dive in pretty rapidly if you sign up for one of these courses. But the hardest part is really intermediate learners, and that's where I feel I am. I'm competent enough where I can do what I need to do, but I'm not happy with my progress. I really want to become fluent in more languages. I really just need time and dedication to keep improving, and there's not really training courses for that. And there's also not the time for that, unfortunately, as you go on and on in your career. So I worry a little bit about my generation who's coming into bioinformatics without the formal training at the university level. And I want to keep up. I want to stay with the state of the art, but it can be really hard coming from that biological background. All right. And the final point I wanted to make too is that we have fundamentally changed the way in which we're analyzing data from a biological perspective. So going back to the taxonomy, we've gone from actual images of things that we can look at and we can manipulate and physically move around to a completely different data format, which is entirely text-based. And this shift is, it's good because it means we can look at more. We can look at more data. We can look at a wider swatch of taxonomy. We can look at more parts of the genome. But sometimes it's good to have a narrow view. Sometimes it's good to hone in on taxon-specific information. And when you're shifting from images to text, even if you have that taxon-specific knowledge, it doesn't necessarily help you analyze your data better. So I can put up nematode taxon names, the nematode genre. And because I have a taxonomic background, I can tell you that, these different genera really tell me different things. So immediately when I see this text, I'm getting information about the pictures which that represents. And I can tell you about the feeding ecology of nematodes or symbionts, or in the case of C. elegans, contamination in my data set. Now, as a biologist moving to these environmental sequencing approaches where we're capturing a wider breadth of taxonomy, this is also a problem because I'm getting a lot more names than what I know. So these are examples from the bacterial world. And when I see these names, because I don't have a formal microbiology background, I just don't really know what that implies from my data sets if I see these things. So, I want to make a case for bringing back images into the way that we're analyzing these data. And I don't say that we need to go back to taxonomy and bring back taxonomic images, but we really need ways which we can represent these text-based strings as visual images, so shapes and colors and patterns, and really use our innate human abilities. So our brain is the result of millions of years of evolution, and we're really good at picking out patterns and spotting outliers. And I think we need to take advantage of that more. And so part of my work, and this is where the open source part of my research comes in, is developing data visualization frameworks for environmental sequence data to help biologists deal with this data and help them very quickly use their expertise to spot trends and outliers and patterns in their data sets, but also to get data visualization as something that we do as a normal part of research. So, I think that research shouldn't always be hard. As a biologist, we're used to having things be really hard and really time consuming, and it's not science if it's not hard. But sometimes we should just be able to upload a file and look at patterns, and it should only take 30 seconds. Some things don't have to be hard. And so this is a project funded by the Alfred P. Sloan Foundation, and it's called Finch. So it's accessible in Google Chrome. If you go to that website, it's up and running now. And this is a framework where you can take the outputs of something like the QIIME pipeline. So we're using biome-formatted files, which is a JSON-formatted file that's a computationally standard format. And it can accept anything from an OTU table to metagenome data, gene variant data. It's really flexible, and you can basically put whatever type of data you want in there with associated environmental metadata, information about your samples. And in a couple seconds, you can just have it up and running and explore your data in a visual sense. So we tried to make this specifically easy. This was done in conjunction with the Data Visualization Studio. So they were the ones that were engineering the software. It wasn't biologists, because we don't always design very good software. So essentially, you're just using a single file, uploading it into this web tool. It's written in CoffeeScript, so everything in the file is detected automatically and uploaded into this parser window. So these are all the samples that you might have. You have metadata associated with those samples, so if you want to narrow down the scope of what you're looking at, in this case ammonia, these are samples from an aquarium at UC Davis. You can do that. And all your samples will be updated in real time. So this is kind of similar to something like kayak.com, like flight websites, where you have those slider bars and you can immediately manipulate your conditions and just have the information updated in real time. And right now we have 5 visualizations that are up and running. So this is a prototype framework, very much so. But the idea is really to to give data to biologists in a way that they wouldn't be used to looking at it. So in this case, representing species as shapes. So size corresponds to abundance in the sample. Having things like search boxes up here. So if you're interested in a specific taxon, you can just search for it. It'll autocomplete, and then you can find that in your data set. And then get an idea of how that taxon is distributed across your samples, and start to use your biological expertise and say, that's interesting. Maybe I should explore that further. So there's things like also taxonomy slider bars. So a lot of times in bioinformatics software, if you want to look at things in an overview at the phylum level versus lower level, like genus level taxonomy, you might have to go to the command line and regenerate your files and have a lot of different files. But we're really trying to make it easy for people just to swap between levels, because a lot of times as a biologist, you're interested in both the higher level and the lower level patterns. And you need to kind of think about those in conjunction with each other to make interpretations about your data set. So we're also using new ways of visualizing data. This is coming from the data visualization world. So this is a Sankey diagram, which I'd never heard of until the Data Visualization Studio implemented it in the software. And this is just looking at the breakdown of your OTUs across taxonomy. So going from kingdom to phylum to class to order, and looking at how that breaks down. So you can see that sometimes you have a couple minor phyla that are represented in your data set, but you might have the majority belonging to a larger group. So this is important when you have a specific sample set. You're oftentimes you want to know how things break down and what is the most abundant versus the rarer tax in your data set. And the other thing is interaction. So there's surprisingly few tools where you can really interact with your data, where you can just toggle between levels, where you can switch between numbers of sequence reads and percent abundances, where you can hover over things and get more information about what that sample represents. So, the taxonomy, the percent, the number of sequence reads in the sample, things like that. So in designing this open source tool, we're really trying to make it easy for people to interact with their data, because that interaction is really important. And that's something that I think we've lost a little bit coming from biology to bioinformatics, because I am used to physically sitting under the microscope and manipulating my samples and going up and down the focal plane and rotating my specimen to kind of make decisions about what I'm seeing. And when you're dealing with a text file, it's very difficult to do that, because you're kind of just staring at the text file and you don't really know what you should be doing with it. You can run a couple scripts, but it's hard to write an algorithm for something that you're not expecting to see. So really, data visualization is a way for us to explore and discover these patterns which might not be obvious in our data sets, and look across a wide swath of taxonomy. And this also has implications for citizen science and outreach. So one of the things that I'm also interested in is in getting these really exciting genomic data sets out to people that are interested in science, but they don't want to use the command line. They don't want to download bioinformatics software. So part of what we've been doing also is working with citizen science projects like Wildlife of Our Homes, where people actually sampled their homes and sent in swabs for sequencing. And then when they get their data back, instead of getting a text file, they can actually just go to the website and explore the data sets in the visualization framework. And they're excited about this because they know their samples are in there. So maybe they'd be able to discover interesting patterns themselves without actually having to have that scientific background. All right, so this is a prototype framework. I'm happy to talk more about this, but in the next phase, I want to expand this. I want to keep working and moving into the data visualization world, because I'm really excited about it, and it's been a really fun experience having this project funded. My background's phylogenetics, so I want to incorporate phylogenetic visualizations into the next phase, develop statistical tools, plugins, public APIs so that other people can interact with this. The source code's on GitHub. It's fully open source, and I'm happy to talk more later. But in the end, I really would like to see a self-sustaining open source user and developer community spring up around these type of visualization software. All right, so I'll just summarize here and give some concluding remarks about the future of biology and bioinformatics. Again, I'd love to hear comments and discussion after this talk, and hear some responses. But I would like to see more interdisciplinary training, collaboration, interaction, especially for to prevent a lost generation. People like me who are new PIs, which are coming into this world, and we don't have the formal training in bioinformatics, but it's going to be really important in our careers to adapt these tools and to use genomics and sequencing and to stay competitive. I'd also like to see bioinformatics tools that take advantage of taxon-specific biological expertise. So something like in a tool, maybe pulling in information from other sources, taxonomic databases, GenBank, just giving us more information so that we can actually use our expertise, which is very hard won, to interpret these data sets. And then also new frameworks that also reduce the need for taxon-specific expertise. And by this, I really mean things like data visualization frameworks that allow us to see shapes and colors and patterns and not necessarily know what the genus name or species name is for a particular thing. So with that, I'll finish up and I'll take questions, and just thanks again for inviting me.